Don't worry, Kinman. Just the third time you've done this video. Surely all your kids will watch it. I have not watched your videos, Mr. Kinman. Did you not just hear me say that this is the third time that I've done this video and you haven't watched it? I really don't care about you and your videos. Okay, I understand. All right, guys, so this is Unit 2. Congratulations, you've made it to Unit 2. You've taken the test by this point. Some of you are probably sitting in the class with me right now. Others of you are at home, and I will see you tomorrow. So we're going to go ahead and get started talking about Unit 2. Unit 2 is about cells. So the question that is on your mind right now is, what are cells? Can you answer that question just from your own brain? Cells make up everything. They're the structural units of all life. I just read that from the notes, but it was also in my brain. Some organisms are made up of only one cell, which is unicellular. Some are made up of many cells, like me. I'm made up of trillions of cells. I am multicellular. Now, that said, there are two different types of cells. We have prokaryotic cells and we have eukaryotic cells. Go ahead and ask the question, which one am I? Eukaryotic. You are eukaryotic. Now the prefix eu, EU, doesn't really mean that you. It means another you. So it's not you, it's you as in true. Anyways, prokaryote. Just what are prokaryotes? So pro prefix means before. What does kary, K-A-R-Y? What does kary stand for? Nucleus. Before nucleus, cells. So these types of cells came along or do not have a nucleus. So it says in your notes that they have no membrane-bound organelles, like lysosomes, like the nuclear membrane, like the Golgi apparatus, like the endoplasmic reticulum, rough or smooth, mitochondria. All these organelles, prokaryotic cells do not. They are, in a simple term, they are more simple, less complex than eukaryotic cells. In your notes, we have a diagram, so let's look at it. So this is a diagram of a bacterial cell structure. Notice we have ribosomes, these little dots all inside. Ribosomes function to make proteins. We have pili, which is plural for pilus. Pili can be used in sexual reproduction and locomotion. We have DNA, so bacteria have their own DNA, but they actually have their DNA plus extra DNA, which we'll talk about further later. Some of them have flagellum, not all of them do, but these flagellum whip around little tails that make them move along. Some of them have capsules for extra protection. Within that, they have cell walls similar to plant cells, but made of a different substance. Peptidoglycan is what it's made of, not cellulose like in plant cell walls. And inside that, just like us, they have a plasma membrane. Okay, so it is believed that the first bacteria came into existence approximately 3.5 billion years ago. And the evidence for that time frame are the prokaryotic fossils found in what are called stromatolites, which are bacterial mounds found in sharp clay. So I have here a little picture of that. So this is a picture of several stromatolites, which basically look like giant rocks. However, in further investigation, you actually see fossils of prokaryotes. Specifically, these would be cyanobacteria. Cyanobacteria are special in that they can undergo photosynthesis, taking light, converting it into energy, just like plants do. So this process of photosynthesis also creates calcium deposits, which help build these mounds. But these bacteria also form mucus-like substance that catches sediment and helps to build the structure. So our next cell type are eukaryotic cells. Now remember, I said eu as in Y-O-U. Those U are eukaryotic. But EU, the prefix, actually stands for true, as you see here in your notes. So what does that mean? True carry, which means nucleus, so true nucleus. So these types of cells have nuclei, and they also have many other membrane-bound organelles. In other words, structures inside that are contained within a membrane. So that is an important distinction to make because prokaryotic cells do not have organelles. They are smaller, simpler than eukaryotic cells. So where did those organelles come from? Well, that leads us into the topic of the endosymbiotic hypothesis proposed by Margulis in the 60s. He proposed that some of the organelles found within eukaryotes came from prokaryotes that eventually started living inside of the bigger eukaryote. So as we see here in this diagram for the endosymbiotic theory, you have a gradual progression into a more complex type of cell, which eventually your end result is your modern eukaryotes. So here we have a modern photosynthetic eukaryote, like what you would find in plants. Here is a modern heterotrophic eukaryote, like you would find inside of your body that you're made of. So it started with a simple proto-eukaryote. That prokaryote started to fold its membrane inward to compartmentalize things that it was taking in. Now understand that this is a small diagram, but this must have occurred over millions of years. All right, and that is not represented in this diagram, but make sure you understand millions of years is thought to have occurred before all of these events happened. Eventually, the proto-eukaryotes formed a nucleus, becoming more like the eukaryotes we see today, forming an endoplasmic reticulum. And if we actually look at the structure of the membranes of these parts, the structure is actually just like, for the most part, the cell membrane. And so then these larger eukaryotes lived in close relationship with smaller aerobic bacteria. And as you see here, the bigger one is engulfing the smaller one, but look what happens. It does not break down and digest this small guy, because this small guy, this prokaryote, is aerobic. So it's taking in oxygen and converting that into energy. So it begins to feed the bigger one. So the bigger one is getting nutrients from the smaller one, and therefore does not digest the smaller one, just keeps getting energy from the smaller one. Now the smaller one is better protected now that it is inside this bigger structure. Same thing for this diagram. We have our aerobic bacteria that give rise to our mitochondria, but then we have a photosynthetic bacterium, kind of like the cyanobacteria that we talked about before, could photosynthesize light into energy, and then they begin living in a symbiotic relationship and become the plant cells that we see today. So ask yourself the question, why did this happen? Why did the small ones start to live with the big ones, and the big ones allow the small ones to live inside of them? Well, as it says here in your notes, the smaller ones gained protection, the larger ones gained energy for faster motility. So they became overall, became a bigger system, more likely to survive, therefore pass on their genetics to create future generations that resemble them, which is what evolution really is. Over time, DNA segments from the smaller ones and the bigger ones began to swap, creating a more permanent existence, which is called genetic annealing, taking genes from one, stitching it into genes to another, and making it a part of the system. All right, so here's an important question to ask. What is the evidence of this? What is the evidence found in mitochondria and chloroplasts today that support this theory? So mitochondria and chloroplasts have their own single circular chromosome like bacteria. So this is very important. Bacterial DNA is not like our DNA. Bacterial DNA is circular. Like what you find in mitochondria and chloroplasts inside of your own cells right now. They have their own DNA that looks just like that of prokaryotes, bacteria. So that is one piece of evidence. Your DNA is linear. It's a line. It has a beginning and an end, but not bacterial DNA. It is circular. The next piece of evidence is that mitochondria and chloroplasts have their own ribosomes. If you remember earlier in this video, one of the key characteristics of a prokaryote is that it has ribosomes. So do mitochondria and chloroplasts. The next piece of evidence is that mitochondria and chloroplasts are able to independently reproduce within the larger eukaryotic cells. They use a similar process to binary fission. So they start off at one cell and they split into two cells. Inside of our cell, they're doing this. And so that is another piece of evidence that supports this endosymbiotic theory. So I want to finish up this video with comparisons between eukaryotic and prokaryotic cells. This is really important that you graduate this class understanding this difference. I would feel like a failure if you did not understand what a prokaryote, what a eukaryote were. So look at this diagram. Yeah, so don't fail me. That's what you're thinking, right? This diagram shows plant cells and animal cells here about 100 micrometers. So these we can see under a microscope, and you are hopefully going to get to see them under a microscope whenever you come to class, or the next day you're in class right now, or you may not be. Anyways, and here you have bacterial cells. Notice we go from about 100 micrometers down to about 1 micrometer. So prokaryotic cells are a lot smaller, and then even smaller than that we get to viruses, which are only about 100 nanometers, and then we get even smaller all the way down to atoms. But the thing to see here are the difference between bacterial cells and eukaryotic. They're way smaller. All right, and here's our diagram for bacterial structure again. And the next thing it talks about here are the 3 different shapes of bacteria that you could find. So we have cocci, which means round. We have bacilli, which means rod. We have helical, which means spiral. And in this diagram, we have the different bacterial shapes. We have pictures of the 3 shapes we just talked about. We have coccus, which is singular for cocci. An example would be diplococci. And then we further add, it's like adding beads to a chain. Then we have streptococci. So strep. If you ever had a strep throat, then maybe you have a contamination of these bacteria. Or if you've ever had a staph infection, you have staphylococci, Staphylococcus aureus, staph infection. Then you have your bacillus shape, which are shaped like rods. And then you have your spirillum, also known as helical because of the spiral shape. So those are your 3 different shapes of bacteria. So back to the notes. Here are your diagrams for animal cells and plant cells. Hopefully you notice there's a lot more going on inside of animal cells and plant cells. Remember, these are way bigger. They are thought to have evolved about 2.7 billion years ago instead of 3.5, larger and way more complex. So hopefully some of these things, if you reminisce, you remember learning about some of these things. All right, so that's the end of this video.